Hello viewers, today we are going to discuss about that circular dichroism spectroscopy. Circular dichroism spectroscopy is based on the differential absorptions of left hand and right-handed circularly polarized light by serial molecules. Serial molecules like proteins, nucleic acid, and some natural products. That is, observe left and right circularly polarized light to different extent produce a CD signal. The CD spectra arise due to electronic transitions for UV region 190 to 250 nanometer. So it is used to secondary structure of proteins like alpha helix, beta sheet, random coil. Near UV regions, the range between 250 to 300 nanometers, it is used to tertiary structure of proteins. For example, aromatic amino acid, disulfide bond, etc. In visible regions less than 300 nanometer, it is a metalloproteins, grumospore and plant pigments. Next, we are going to discuss about instrumentation. The instrumentation includes first one, light sources. The light sources, xenon arc lamp or deuterium lamp, that is UV lamp for broad wavelength range. And the second one, monochromator. Select the monochromator, the specific wavelength of light for measurement. And the third one, polarizer. It is produce the linearly polarized light. And the next one, photoelastic modulator, that is PEM. The PEM is converted linearly polarized light into alternating left and right circularly polarized light. Next, we'll move on to the PEM. The sample holder, that is cuvette, will be there. It is usually quartex cuvettes. The samples must be optionally transparent in the measured range. Next, a detector. It is mainly used for photo multiplier tube, that is PMT, or photodiode. Records the difference in absorbance between left and right circularly polarized light. Finally, the computer and data processing. It is converted the raw signals into the CD spectrum. So here we can see the picture. The light source will pass it through the light, that is white unpolarized light will enter into the monochromator. We go for all the sides. Then monochromator, they can convert the polarized light, that is the only one direction we can move on to that lights. Then monochromator will enter into the photo detector modulator, that is PEM. So here we can convert it into the right circular polarized right and left circular polarized light. Then this both light will enter into the sample, that is the samples will be there in the cuvette. We can take a 3/4 level the cuvette. It is a very clean and coloring solutions, we can take that. After the samples, the light will pass, then enter into the detector. The detectors they capture the signals and we can show it into our laptop or systems. Finally, we take the reading as well as graphs. These are the overview about that instrumentations of CD. So the next, the main important that what are the applications? Where will they used for the CD spectroscopy? The first one we can use biochemistry and structural biology. So here the many laboratory they can use to for main the three important isolate and elute the samples like protein, DNA and RNA. So here the protein have the four types will be their primary, secondary, tertiary, quaternary structure. So here the protein secondary structure determination we can use especially for alpha helix, beta sheet, random coil content etc. And also we can monitoring the folding and unfolding or denaturations or renaturations or stability. The protein folding means if you are uh, adding means SDS, it is an anionic detergent. That detergent, the unfolded protein, they can convert it into linear position. So yeah, the basically generally the protein, if you are go for isolate the protein samples, especially for we can done both qualitative and quantitative analysis. After that, we'll move on to the estimation of proteins for Laudis method or Barford method or Benedict method or anything else. We can use the find out how much amount of proteins will be there. Then after that, we go for SDS. So we can SDS page only before the SDS, the protein will be folded. Then if you are adding the SDS, the protein will be linear form. That purpose only we can use to for the SDS. So mainly the we can find out the with the help of SDS protein structure and molecular weight we can use to for it. And the next one, analysis of nucleic acid conformation, that is A or B or Z DNA or RNA structure. Next one, the ligand proteins or drug DNA interaction studies also used for CD spectroscopy. So the next, material science and chemistry, studying the shearlities of organic and inorganic substance or complex, and also the characterizations of chiral catalytic analysis, especially for the catalytic means the lock and key model, ligand receptors and the substrate enzymes we can need, that is the purpose of catalytics. And also nanoparticles or nanomaterials we can use the characteristic purpose of CD. Next, the analysis of polysaccharides and synthetic polymers. And the next, where will the use of CD spectroscopy means the pharmaceutical as well as biochemical or biomedical applications. So here the quality control of biopharmaceuticals, that is a therapeutic protein and vaccination. We can particular vaccination how much amount of quality, it is a quality control we can find out that purpose we can use the spectroscopy. Apart from that, screening for endomeric purity of the drugs. What are the meaning of purity of drugs? How much amount that is purity? Like that we can find out. After that, studying the conformational changes in disease-related protein, like amyloid and pyridine, etc. So that purpose we can find out more or less in the CD spectroscopy used for the pharmaceutical as well as biomedical applications. Next, what are the advantages? What are the advantages of a CD? So, the main function, structural informations on biomolecules. So, we can find out the protein alpha helix or beta sheet or random coil, which one the types will be there, like that we can find out. And no distractive techniques. It is for samples can be recovered after the analysis. Next is the small sample requirement. We can need milligrams amount of protein. It is enough for that. Otherwise, nucleic acid also. Fast and simple. Okay, it is easily quick and compared to the crystallography or NMR, and also sensitive to conformational changes. If you are folding or unfolding or monitoring like that, or stability like that, denaturations of proteins or denaturations of DNA or RNA like that, so sensitivity to conformational changes. Apart from that, applicable to a variety of biomolecule, that is proteins, peptides, nucleic acid, chiral organic compounds like that. Next, in-solution measurement, like to analyze the biochemicals near or native physiological conditions like buffer pH and temperature. And last one, the most advantages of CD spectrum, quantitative secondary structure estimations like that is we can used for the computational analysis of spectra, can estimate the proportions of alpha helix and the beta shade. These are the so many advantages will be there. Apart from that, disadvantages also will be there, like low resolution power, that is cannot provide the automatic level structural details like X-ray crystallography or NMR, require chiral samples like only the optionally actives or molecule produce useful for CD signals. Next, a limited wavelength range in aqueous solutions, like the water absorbs strongly below 190 nanometer and restricting for UV measurements. Buffer interference, like many buffers, salt or additives absorb in the for UV, which can distort spectra, require careful calibration, that is air artifact, that is for from light scattered into the tuberite or aggregated samples, can be affect for the accuracy. Next, the not suitable for very complex measure, that is overlapping the signals from multiple component may be complicated interpretation. And weaker signals compared to the absorption spectrum. The CD signals are small, like that at 10 power minus 3 to 10 power 6 of absorption intensity, require it is a sensitive instrumentation. So now we are discuss about that what is the principle as well as instrumentation and application, advantage and the disadvantages of circular dichroism spectroscopy. I hope you can understood like. Thank you so much for your listening. Thanks a lot.